Before lab, please review the ASTM standard available on the course Canvas site. We highly recommend the students utilize the standard to create a form which will streamline data collection and ensure all required data is recorded. Before testing a liquid fuel, we need to standardize the calorimeter using benzoic acid, which has a known heat of combustion. Obtain a clean metal crucible and measure and record the initial mass. Open the jar of benzoic acid, extract one pellet, and deposit in the crucible. Now measure the combined mass of the crucible and pellet. The net mass of the pellet should be around 1 gram. The sample preparation of a liquid fuel is more involved than the benzoic acid. As previous, the mass of the empty crucible is measured and recorded. We'll use cellophane tape to seal the crucible chamber to prevent evaporation of the fuel. Unroll a 2 to 3 inch strip of cellophane tape on the table with the sticky side upward. Please double back the tape for the next person to use. Now invert the crucible and firmly press the rim onto the tape, then use an X-Acto knife to trim the excess tape along the rim. Next, cut a 3/8 inch by 1 inch strip of tape. Apply the tape flap to the membrane and pull back halfway. Reweigh the crucible to determine the mass of the cellophane tape. Measure and record the mass of an empty syringe. Open the storage jar and draw out the predetermined volume of fuel. Afterwards, please return the fuel cap. Measure and record the mass of the fuel-filled syringe. Pull back the tape flap and inject the fuel into the sealed crucible using the syringe. Now cover the injection hole with the tape flap. As the sanity check, measure the mass of the fuel-filled crucible. Place the crucible in the electrode ring of the bomb head. Next, use the scissors to cut a 10 centimeter length of fuse wire. Carefully install the fuse wire between the two quick grips electrodes by lifting the cap and threading the wire through the eyelet. Once threaded, lower the electrode cap. Gently adjust the fuse wire to loop over the pellet without actually contacting either the pellet or the crucible. If using a fuel sample, prick the membrane with a needle to prevent collapse during pressurization. At this point, we're ready to close the bomb vessel. Open the outlet valve to allow air to vent during assembly. Slide the bomb head into the cylinder and push down until fully seated. Now carefully thread the ring cap onto the cylinder. The ring should easily thread. If not, it's possible it's cross-threaded. When properly closed, no threads on the cylinder should be exposed. The bomb vessel is pressurized with pure oxygen to facilitate combustion. Gently close the outlet valve of the bomb head. Next, slide the connector head onto the inlet valve and slide it down until fully seated. Now crack the main valve on the oxygen tank a quarter turn max and slowly open the filling control valve and allow the bomb vessel to pressurize to 30 atmospheres. Close the filling valve and release the residual pressure within the filling line by depressing the relief valve. The pressure should now be 0. Remove the connector head from the inlet valve and attach the handle. The goal is to fill the water bucket with 2,000 grams of water within a half a gram. First, measure the mass of the empty bucket. Contrary to the video, the bucket may remain on the scale while adding 2,000 grams of water. The graduated cylinder provides a ballpark volume, Then use an eyedropper to fine-tune the water to the target mass. Use the handle to lower the bucket into the calorimeter. Orientation is critical for it to seat properly. Then lay the handle down. Grasp the bomb handle and partially lower the vessel into the water. While holding the head above water, connect the two ignition wires. Now fully immerse the bomb vessel into the water. If any air bubbles are observed, abort the test and contact your GSI. Otherwise, remove the handle and set it aside. Gently lower the top cover onto the calorimeter with attention to orientation of the thermocouple and pulley. Slip the drive belt onto the pulleys and turn the switch to start the motor. Open the LabVIEW shortcut folder on the desktop and select Thermocouple Log Main. Start LabVIEW by pressing the white arrow along the top menu, then click on the Take Data button to initiate temperature recording. Let the motor run for 5 minutes to allow the water temperature to attain equilibrium before a measured run. At the start of the 6th minute, stand back from the calorimeter and fire the bomb by pressing and holding the ignition button until the indicator light goes out or 5 seconds has expired. Normally, the light will only glow for a half a second. The water temperature should start to rise within 20 seconds of ignition. After several minutes, the temperature curve will start to level out and attain steady state. Click on the Stop button and save the temperature data file to your desktop using a descriptive file name. Turn the switch to stop the motor and remove the drive belt. Lift the cover and place on the provided stand. Connect the bomb handle and partially extract the bomb vessel from the water. Using an eyedropper, remove any excess water from the top of the bomb head, then remove the ignition wires. The bomb vessel may be placed on the workbench at this point and the handle removed. Slowly open the outlet valve to release the gas pressure within the bomb. The venting should proceed slowly over at least a minute to avoid entrainment losses. Once the vessel has fully vented, unscrew the ring clamp and set it aside. Lift the bomb head straight out, no twisting. Now rinse the electrodes and capture all the washings into the bomb vessel before placing on the stand. Wash the crucible and all interior surfaces with a jet of distilled water and collect all the washings into a glass beaker. Recover whatever is left of the fuse wire. Then measure and record the combined wire length. Measure and record the mass of the beaker with the bomb washings. Carefully add a few drops of methyl orange indicator solution to the beaker using an eyedropper while swirling to induce mixing. Typically 2 to 3 drops is sufficient. Note the initial volume of titration solution in the burette. Position the beaker under the burette. Slowly open the stopcock to add titration solution to the beaker while swirling the mixture. Stop when the mixture turns clear. If the mixture turns another color, you've gone too far. Record the final volume of titration solution in the burette. Then calculate the net volume of titration solution used. As a sanity check, you can measure the final mass of the beaker. Before leaving the lab, be sure to save your data.